Hi guys, so we've got another set of excretion questions, so pause the video here, try these questions out and continue on to see how I answer them. Right, C per I is crazy. This question should be illegal. It's that bad. Nobody can get this right because it's beyond me. But you know what, let's see what we can learn in terms of exam technique from it, okay? So just ignore the answer for now, just don't even look at it. Might be hard, but let's just take it as if, we got it in the exam at face value. Right, as I always say, read the question first. Explain why podocytes are usually unable to undergo mitosis. Now, let's break this down. Firstly, the command word is explain. So clearly you know the information. This is a regurgitation in a way. But the problem with this question is that it's saying the podocytes are unable to undergo mitosis. I don't know that, do you? Because we weren't taught that in class, were we? Right, so this is where exam technique comes in. Ignore the podocytes. Who cares about them, right? Cross that out and just change the question to this. Explain why structures, cells or something, are unable to undergo mitosis. And with that, you have an answer to say, okay? Very easily, well, if they're not undergoing mitosis, they're specialized, right? If they're specialized, they're in genes like G0, okay? Why can't they undergo mitosis? Well, what are the criteria? What are the requirements to be present for mitosis to occur? Well, let's say the cytoskeleton needs to function, or the spindle fibers need to be formed. Do you get my point? And that's 3 marks by saying those 3 points. So, if we go back, the only point that I haven't mentioned is this. The shape is too irregular. Like, guys, I wouldn't get this in the exam, right? There's no way I'm going to say that. But I learned it from this question. And they gave, to be fair, they did give you the diagram for a reason, right? But we didn't need to use the diagram because we still would have gotten the 3 marks. But this is a learning lesson. Write this in your notes that mitosis can't occur if the shape is too irregular. Clearly, we can see the podocytes have loads of projections, right? These things just going out, right? That's too irregular. It's asymmetrical, so it can't do mitosis. And that's how you answer the question. Right, moving on. What do we have here? So state and explain one symptom you would expect to observe as a result of that sort of diabetes, whatever. Okay, I don't know anything about that sort of diabetes, so I'm going to read. Okay, so it's a disease of the kidney that affects the regulation of water potential in the blood. Remember, they say, the examiners say things for a reason, so that's something very important. So one cause is lithium poisoning, and lithium ions enter the kidney tubules through sodium channels. Now, this is important. This prevents the cells of the collecting duct from responding to ADH in the blood. Okay. What would we expect as a symptom? Remember, symptoms are like a fever, a rash, thirst. Those are symptoms, right? So don't get that twisted. So if it is preventing the cells of the collecting duct from responding to ADH, well, then clearly the problem we have over here is the fact that there will be no aquaporins being inserted into the cell membrane of the collecting duct. So if there is none or less of them, let's just say, right, then, well, there's going to be all that water being taken, excreted out of the body as urine. So if there's water loss from the body, well, clearly you're going to be thirsty, right? You're going to be dehydrated, and that's how you would answer the question. So what's really important is identifying what's the main topic, water potential, okay, but what's actually causing the water potential to change is the fact that ADH can't actually, get there into the collecting duct. So this is linking your knowledge of aquaporins to ADH to water potential. Then lastly, is the cute question, which letter or letters label areas of the nephron which are directly affected by ADH? Easy, collecting duct, M. Okay? So that's pretty much it. I hope this video was useful. If you have any questions, let me know, and I will see you in the next video. Bye bye.